molecule to a therapeutic agent from design and synthesis to in vitro and in vivo studies against Parkinson disease. We are very happy to have one of our previous students, Dr. Moumita Jash, Associate Scientist, GVK Bioscience, Aragen Life Science CRO, Bangalore. To start with the invited lecture, I would now like to request our principal sir to please share his views on the same. Sir, please.
Good evening to you all. Thank you, Nivedita Madam. Welcome, Dr. Moumita Josh, on of our alumnus in the Department of Chemistry. Despite your busy schedule, you have agreed to give a wonderful lecture on the subject matter as mentioned by Nivedita Madam. And the students, you are very lucky enough because you not only you will gather some information knowledge from ma'am, but also you should know how I will request madam to please share some words how from your journey, that means from Durgapur Government College to then post graduation, then PhD, now your present position. Please share at least 10 minutes to our students so that they can be motivated and they should know that if you continuously do your best version, so time will come and you will be established in a reputed organization, institution, university, whatever may be. So, and I must thank to head of the department, Snigdha ma'am, other colleagues. So not only it is beneficial to the student, but also the faculty members. You can make a collaboration with madam and gather some information. What the research is going on this particular subject, particularly the Parkinson disease is very important, and old aged persons are there. This is the burning problem throughout the globe, and how to combat the particular disease. So we must learn something from madam, and as well as we can make a collaboration with these few words. Once again, my as in all invited lecture, as this is my usual practice to invite you in the physical mode because it is your college. So, Moumita madam, please, whenever you come into Calcutta or Durgapur, whatever, please visit our college and motivate our student, not only the student but also the faculty. It is the alma mater. So, with these few words, I wish a grand success of the seminars. And dear student, please interact with your students. And DBT has opened up a new dimension to interact with such eminent resource person before you. Thank you. Thank you so much.
Yes, sir. Many thanks for your kind words. You have always been our inspiration behind organizing all of the events. Thank you, sir. Thank you very much for joining us. So, to start with the technical session, I would now like to request our head of the department, Dr. Srikda Chandra, to please introduce the resource person. Ma'am, please.
Okay, thank you, Nivita. Thank you, sir, for being with us all the time and supporting us. Good evening, everyone. This evening, I have the unique opportunity to introduce Dr. Moumita Josh, Associate Scientist, GVK Biosciences, Bangalore. We are so happy and proud to have you, Moumita, with us. Moumita has born and brought up in Durgapur. She has completed BSc from Durgapur Government College itself in the year of 2011 and Masters from Badon University with the specialization in organic chemistry in 2013. Then she joined the PhD program in Indian Institute of Chemical Biology under the supervision of Dr. Chima Chaudhary after qualifying NET and GATE in 2013. In her doctoral studies, she was involved in the synthesis of numerous biological catalyzed heteroannulation reactions. She earned her PhD degree in the year 2020. Following that, she received the National Postdoctoral Fellowship and joined the Joint Professor Surajit Ghosh's group in January 2021, where she designed and synthesized various active scaffolds against cancer, Alzheimer's disease, Parkinson's disease, and stroke. Currently, as I already mentioned, she is employed as an associate scientist in GVK Biosciences, Bangalore. My heartfelt thanks to Moumita from all of us at Durgapur Government College. Thanks for taking out time for your juniors. Your journey will motivate our students who are with us now. So without taking much time, I would request Moumita to start her talk.
Thank you so much, ma'am, for your kind words. It's really a pleasure to give my presentation in front of so many eminent teachers. And I would also like to thank the principal of Durgapur Government College, Dr. Palik, for giving me an opportunity to present my work here. With this, I would like to start my presentation. The journey of an organic molecule to a therapeutic agent from its design and synthesis to its biological studies against Parkinson's disease. Now, for a molecule to become a drug, there are many steps which are involved for a molecule to be a drug. For example, paracetamol we take when we have fever. But although the molecule is very simple, but it has not become a drug in a single day. A lot of steps are involved for a simple organic molecule to become therapeutically active. So here I will first say the sequential steps which are involved for designing a new therapeutic molecule against any particular disease. The first step which is required for designing the molecule is a very thorough review of literature. Now, a thorough review of literature of any particular disease which we are going to work with will give us a number of drugs as well as many therapeutic molecules which are involved in that particular disease. But if we look into the close structure of those molecules, we will see that there are many demerits as well as limitations which are associated with those molecules. Now, because of these demerits or side effects, these molecules cannot go into further study. So, as an organic chemist, our work is to find out that which structural moiety, which functional group or which side chain is responsible for those demerits or limitations of those molecules. Now, and then design new therapeutic molecule based on those basic scaffold. Now, designing of this molecule involves a large number of molecules. For example, um, innumerable number of molecules can be designed from one single molecule. But in a lab, for a single particular study, it is impossible for anyone to synthesize all those molecules and do a biological study. For this, we take help of computational method. In these methods, is the second point of designing molecule is computational method where we use software which can predict the binding ability of a particular molecule with a protein which is responsible for a particular disease, and this binding affinity or ability will give a docking score based on which we can say that which particular molecules are can be an suitable active agent or which one we can eliminate from our study. After having designed this molecule, our next work is the synthesis of the designing molecule. This involves four basic steps. One is the retrosynthetic analysis. Second is the synthetic synthesis of the molecule using either new methodologies or modifying existing methodologies. Third is the purification of the molecule because no biological work we cannot take our molecules into biological work if it is not more than 95% pure. So purification of the synthesized molecule is a very vital stage. And fourth is the characterization of the molecule. Whichever molecule we have designed should be well characterized so that we can identify that this is our design molecule. And in the and next, after designing and synthesis of the molecule comes the biological evaluation. Now, the most important study for biological evaluation is cytotoxicity study, where we should first see whether our molecule, when it enters into biological system, whether it be a cell or an animal, it should be less toxic. That means the cell should be viable. The molecule should not kill the cell or the animal. So cytotoxic study is a very important parameter for and for biological analysis. After cytotoxic study, after the molecule passes the cytotoxic study, we can come to in vitro assay, which is the outside cell assay, or in vivo assay, which is the assays on living organisms. Now, these are the steps which are involved for designing and synthesis of a therapeutic molecule. Now, today we are going to talk about Parkinson's disease. Now, first we should know what is a Parkinson's disease. It is a progressive movement disorder of the nervous system where different parts of the brain cells, especially in the substantia nigra parts, they become weakened or damaged. And basically, these nerve cells or neurons are responsible for producing a chemical messenger named dopamine, which is responsible for movement. So, in Parkinson disease, the symptoms mainly is tremor, that is shaking of the body, rigidity, which is muscle stiffness, bradykinesia, which is the slow movement of spontaneous and automatic movement, and postural instability, that is the inability to walk, stand, or sit properly or complete day-to-day daily tasks. Next, coming to the literature survey, the literature survey shows that the only current pharmacotherapy which is available is L-DOPA. L-DOPA, which mainly focuses on the symptomatic relief of the Parkinson disease. That is, the function of L-DOPA is that it will enter into our body, it will cross the blood-brain barrier, and it will break down into dopamine, which thereby altering the amount of dopamine in the nerve cells. That is, it is only causing symptomatic relief of Parkinson disease. It is not responsible for reviving the damaged or weakened nerve cells. So obviously there are definite treatment which should be further progress should be made on treatment for designing of new therapeutic molecule against Parkinson disease. Again, survey of the literature shows that leonurine, which is a primary alkaloid, which is a natural product and which shows diverse range of biological activity and has significant therapeutic effects in cardiovascular and central nervous system. However, it has number of limitations because if we look into the structure of this leonurine, you will find that it consists of two main polar groups. One is this guanidine part and another is the hydroxyl part. And due to this polar group, it restricts its use as a therapeutic agent because of its low viability, weak transmembrane ability, and poor fat solubility. So we decided that if we modify this particular structure of leonurine and replace these groups, that is, these polar groups like guanidine and hydroxyl group, with some other neuroprotective group, then we can definitely end up with a more active therapeutic agent against Parkinson disease. Now this is our design molecule where we have replaced this guanidine part with numerous aromatic aliphatic neuroprotective amines, and this hydroxyl group has been replaced with ester. This has been esterified with different acids. Now this structure is modified form because here the polarity is to much extent reduced. So with this structure we have created almost 20 molecules, and then these 20 molecules are we have predicted we have done the docking study with these 20 molecules. Now the ligand which we have used is that is dopamine transporter, and we have seen that out of 20 molecules, 10 molecules gives a good docking score, and the other molecules the docking score is not to some extent good. So we have taken only those 10 molecules which shows a good docking score, and we have planned to synthesize only those 10 molecules. Next, coming to the synthesis of the molecule, the first part is the retrosynthetic analysis. Now, when we have done retrosynthetic analysis of our design molecule, we have seen that the simplest starting material is a commercially available serine. Next, we started the synthesis of this our design molecule with this commercially available simple serine. This is the synthetic scheme which we have made for the synthesis of our design molecule. First is the Wittig reaction for the homologation of the aldehyde. Now, to perform the Wittig reaction, first step was to protect the free OH group, which can interfere in the Wittig reaction. So, the OH group in the first step was protected with the TBS group, and then Wittig reaction was carried out for the homologation of the aldehyde to an alpha beta unsaturated ester. The next step is to reduce this double bond. Now, there are a number of methods which are reported for the reduction of this double bond, but when we were trying these methods, we were finding that either both the double bonds and the esters both were reducing, or in some cases only the ester was reducing, the double bond was remaining intact. So we needed a milder reagent for the reduction of this only double bond, keeping the ester group intact. And we came out with a method where we used sodium borohydride in the presence of catalytic amount of cobalt chloride, which successfully reduced this double bond, keeping the ester group intact. In the next step, we hydrolyze this ester group followed by this deep protection of the TBS group to produce this molecule 7A. Now, once we got this molecule, we can easily couple it with our design strategy, that is different neuroprotective amines, that is RNH2, which can be aliphatic as well as aromatic group via an acid amine coupling reaction to produce this intermediate A. And after this, the next step was to protect this OH group or esterify this OH group with different acids to produce our design molecule 2A2J. With this scheme, we have synthesized 10 different molecules. And next, after synthesizing this 10 different molecules, our next step will be the this is the 10 different molecules which we have synthesized, having different these each amines which we have used has reported neuroprotective properties. So we have inserted this amine to our design molecule so as to increase its neuroprotective ability against Parkinson's disease, may be better than that of luridine. After synthesizing the molecule, our next step is the purification and characterization of the small molecule. There are basically two main methods of purification. One is the thin layer chromatography, another is the column chromatography. In both the cases, we use silica gel as a stationary substrate, stationary phase substrate, and the separation of the molecules from either the byproducts or other molecule is based on the polarity or hydrophobicity of the molecule. That means as we increase the polarity of the solvent phase, then the molecules get separated based on their polarity or hydrophobicity. Now, after purification of this compound, the next is the analyze the purity of the compound. Using HPLC method, we have analyzed all our compounds, and all our compounds were greater than 95% pure. For example, this was our lead molecule in the study, and it is 97% pure. Similarly, we have analyzed all the molecules, and all the molecules were greater than 95% pure. Next is the characterization of the molecule. Mainly for an organic molecule, there are two methods of characterization. One is through NMR and another is mass spectrometry. Now, this is an NMR spectra of our lead molecule where it shows that all characteristic peaks are present of our design of our synthesized molecule, from which we can say that our molecule has been successfully synthesized. And this is also the mass spectrum, which shows that the mass of our exact mass of our lead molecule is 398. It is showing an M plus H peak of 399. Thus, from this we can conclude that we have successfully synthesized all our molecules, and all the molecules have been purified and characterized through NMR and mass spectrum. Next, coming to the biological study. As we have already said before, the first step is the cell viability study. We have done our study in SHSY5Y cell, where we have used cell viability assay, that is MTT assay. Here we have treated the cells SHY5Y cells with our molecule, and then we have added the MTT reagent, which is a yellow dye tetrazolene reagent. Now, if the cells are viable, then there are certain enzymes present in the cell which will convert this yellow tetrazolene MTT dye to a purple formazan reagent, which will create the purple color. So, if we get purple color, the more purple color we get, the more we can say that the cells are viable. So, by doing the MTT assay of all our synthesized molecule, we found that in SHSY5Y cell, all our molecules are viable. However, some molecules like those which are marked in red, for example 2E, 2G and 2J, those the cell viability values are less than 80%. So these are discriminated from our further studies. We have only taken those molecules which have shown greater than 80% cell viability against SHY5Y cell. Now, those seven molecules then they were carried forward to the next step, that is for the rotenone induced PD model. Now, to generate an in vitro PD model in the cell, we have used rotenone, which is a known neurotoxin which causes PD. And this PD model, and in this generated PD model, we have given our treated with our compound, and we showed that among all the seven molecule, compound 2D shows the maximum neuroprotection, that is 80, almost 82%. And if we see the structure of compound 2D, it is this which has been named as MJ210 as the lead molecule. So further study was carried out with this lead molecule. Now, we have then performed the live date assessment assay. In this, first we have treated the cells with our molecule, that is MJ210, our lead molecule, and then we have treated it with rotenone, that is a neurotoxin which generates PD. Then we have treated the cells with calcium and PI. Calcium, which will stain the live cells green, and PI will stain the dead cells red. We have taken the images, fluorescent microscopic images, and we have seen that more number of cells, when we are using rotenone model cells, then more number of cells are showing red color as compared to that of our molecule treated cells. Here we see that when we treat the molecules with rotenone as well as our cells with rotenone as well as our molecule, we see that more number of green fluorescence is seen, which is telling that more and more number of cells are living as compared to that of rotenone induced PD model. And also when we are seeing only our molecule, treating the cells with our molecule, the number of live cells are still more as compared to that of and comparable to that of control. Thus, we can say that our molecule has neuroprotective properties, and it is recovering the cells from dying. And this is the assay which is showing that our molecule, when our molecule is treated, the cells are treated with our molecule, it is showing a greater neuroprotection as compared to rotenone induced PD model, or even it is comparable to that of control. Next, we are showing the effect of our molecule MJ210 on rotenone induced cellular ROS in SHSY5Y cell. Now, ROS, what is ROS? It is a reactive oxygen species, and it significantly contributes to the pathoprogression of PD, where the ROS molecules can lead to neuroinflammation, mitochondrial impairment, as well as apoptosis. So, by accessing the amount of ROS, we can find out whether our molecule can reduce this ROS generation or not. Now, a dye, that is DCFDA, which is a cell permeable compound, and which when enters into the cell, it causes to convert it into DCF, which is a highly fluorescent dye, and this is caused due to oxidation with ROS. Means when ROS is present in the cell, our dye, that is DCFDA, will convert into DCF, which is highly fluorescent and which we can see through microscopic images. Now, it is seen that when our cells are treated with rotenone, that is the PD model, more and more green fluorescence are seen, that is more and more DCF were produced. That is, it consists of more and more ROS. However, when the cells were treated with our molecule MJ210, it was found that the amount of ROS, the amount of fluorescence is very less, and the amount of ROS has also significantly decreased. Thus, we can say that our molecule is significantly contributing towards the neural protection. So, next, we are going to show the neuroprotective effect of our molecule in rotenone-induced PD rat model. For this in vivo study, we have taken three groups of rats. One is the control group, one is the rotenone-induced PD model, and another is the treated group with our molecule. And then, after the treatment of this rotenone model with our molecule, the rats were sacrificed and Western blot analysis was carried out. And it was found that TH, that is tyrosine hydroxylase, and DAT, that is dopamine transporter, TH, which is responsible for dopamine synthesis, and DAT, which is responsible for dopamine reuptake, both are increasing on treatment of those rats with our molecule. That means this Western blot analysis shows that when the rats were treated with rotenol, the TH and DAT level was expressions were significantly decreasing, whereas when they are treating with our molecule, the levels are significantly increasing. Thus, we can say that it significantly prevented the neuronal death. And this was shown in both the substantia nigra parts as well as the stratum regions of the brain. Next, we are coming to the behavioral study of the behavioral or in vivo study. As we know that Parkinson's disease is mainly associated with motor imbalance or movement imbalance. So we have performed several in vivo behavioral studies with this rats. First is the rotadot test. The rotadot test was used to evaluate motor impairment, balance, and coordination. Now here, the rats were used to sit on a rotating rod, and they were made to balance on the rotating rod. When the rats get disbalanced, that was a measure of motor impairment. And if we see the graph, we will see that when more and more rats are getting motor impairment, that is they couldn't able to balance on that rotating rod when they are treated with rotenol. However, when they are treated with our molecule MJ210, more and more rats are able to balance and coordinate on that rotating rod. Thus, we can say that our molecule is preventing the neuronal disbalance for the rats. Second is the pole test, where which was used to evaluate the motor coordination and bradykinesia, where the rats were made to fall from an 80 centimeter pole, and it was seen that those rotenol-induced rats, they fall very quickly, whereas normal rats, that is the control rats or the rats which were treated with the molecule, were take a lot of time to fall to the ground. This shows that this molecule is also uh, healthy. Next is the breed test. In breed test, we have used the lead of a cage, and then we have made the rat to sit on the lead of the cage, and then slowly inverted it. The rats which were healthy used to grip the cage very tightly, showing that they have good grip strength and coordination. However, the rats which were rotenol-induced the PD generated, they fall or grip very fast. On the other hand, the rats which were treated with our molecule, they could show a good grip strength. Next is the open field state. In open field states, the rats were made to roam about in a new environment, but the rats which were healthy, that is the control, and the treated rats, they explored the whole area of the new environment, whereas the rats which were diseased rat, they only moved only to a particular area or in the corner of a room. This shows that they were diseased rat. Using this behavioral studies and this in vitro and in vivo study, we can say that our molecule, we have successfully designed and synthesized an anti-powerful PD molecule by modifying the structure of leonurine, which was a natural product and which has good neuroprotective properties. Now, this our lead molecule MJ210 thus holds promise to be a potential candidate for developing further anti-PD therapeutics, and we can envision that this molecule has a high potential to be a clinically successful candidate for protecting PD against near future. Now, we have published this paper very recently in European Journal of Medicinal Chemistry, and I would like to acknowledge my supervisor, Professor Shurajit Bose, and lab members, and Sanju Gupta, who is the biologist who has done all the biological data along with me for this work. This was all from my research work. Lastly, I would like to thank all my mentors, although I could not find all the pictures of all the teachers here. Barun sir is missing, although other sirs are also missing. So I could not find in very short period of time. Thank you all of them. Without your help and support, I could not have been where I am today. Thanks a lot, all the teachers. And these are my batchmates, and these are the college memories which I cherish a lot. Although the pictures are very blurred, but the memories are very sharp till now. Thank you.
Yeah, okay, Moumita, it was a really nice lecture, I would say. The amalgamation of all of the things. First of all, the chemistry, which included spectroscopy as well as the biological detection, and I liked most of that, the behavioral model of study which you portrayed. That was very interesting, I would say, and that was very new to me as well, and for a chemist. So many congratulations for what you are doing, and it has been published in a very good journal. So also congratulations for the same. I would now like to request our faculty members or students, if they have any questions for Moumita or any comments, they can please share. Our professors are also there. Borun sir as well as Devashi sir are also there, who were the teachers when you were studying. So they can also share their views, or any comments from the students or any questions are always welcome. Please. Okay, I think students will be having the glimpse of all of the lecture in our YouTube channel. We have recorded the same and we will be sharing it. And for the feedback, I think they will be able to see this. And the most important part is that from the DBT Star College scheme, when you were studying, we didn't had that scheme. Now we have the Star College scheme of our chemistry department, where we can conduct different new experiments, student projects, as well as the thin layer chromatography in the undergraduate level, and also many experiments which are beyond say course curriculum, and also different types of computational studies and workshops which imparts a pre-research methodology among the students. And it was very nice to see our department in your slide, and whenever you get time, I think it was the best one I could see. And this is students, this is the same classroom. I think it is room number 312, I guess.
Yeah, yes, ma'am.
Yes, and we have the same classroom for our students, and it will be so nice if you can visit and you can give a lecture in this room. That will be the best one for our students to share the lecture in the room where you are sitting as one of our students. And I think this is the picture of the exhibition which you
Yeah,
have
yes, ma'am.
portrayed.
Yes, ma'am. Exhibition.
Yes,
College.
yes,
Hmm.
yes. And also, I would like to inform you that our department has now the postgraduate as well as the PhD affiliation. And
Okay.
PhD students are also registered to Department of Chemistry, Durgapur Government College. So
Okay.
this is the advancement I wanted to share with you. So if there are not any questions, so we can connect with Moumita again. We are not ending this session, we are connecting with Moumita, and we have also established the Alumni Connect cell, so that we can remain in connection with our so esteemed alumni. So many thanks to Moumita for sharing her views, and we will be keeping in touch with you. So
Thank you
thanks,
so much,
Okay,
ma'am.
let me
thanks to.
introduce
Yes, ma'am
Nivedita
is
ma'am
there.
in other way. She is the coordinator of DBT Star College team. Okay, departmental as well as central
Ma'am,
coordinator.
I
So
cannot hear
all the
you.
responsibilities with
Yeah,
are with
ma'am, can you
her,
please
and she
repeat?
is conducting the course in such a nice way. Everything. ওকে আমি ছাত্রদেরকে স্টুডেন্টদেরকে বলবো ইউ ক্যান আস্ক এনিথিং ইন বেঙ্গলি অর মৌমিতা যেটা বললেন সেটা তুমি যদি জিস্ট চাও বাংলায় ইউ ক্যান আস্ক ফর শি উইল এক্সপ্লেন ইট ইন বেঙ্গলি অলসো শি ইজ আ বেঙ্গলি গার্ল নো প্রবলেম ওকে বাংলায় শুনতে চাও if you any one of you raise hand or anything or response any response from students কারো কোনো প্রশ্ন বাংলায় করতে পারো এনি ল্যাঙ্গুয়েজ মানে বাংলা ইংরেজি হিন্দি যে কোনো আই থিঙ্ক শি ক্যান স্পিক all the three languages.
Yes, they can also ask questions after that. We will be communicating to Moumita, because then we can just share they can just share their views in the feedback as well. Yes, it
Right,
was.
no problem.
Yes, yes, yes. It was so nice to have you. And the most important is that you have all facilitated all of the undergraduate things which we are doing. Students are doing TLC
Yeah,
now.
yeah, yeah. That's why I have uh, kept all of them in the slide so that they can understand.
Yes, yes. I saw you told about the column chromatography, you told about the thin layer chromatography, or the experiments we are designing for our students. And it is
Yeah,
so nice to see the board where we enter the department. It gives
yeah.
us pride that how our students are doing in the different industries and things. I have one question, whether any human trial being initiated for this?
No, till now not initiated. It's just we
Okay,
have finished the in vivo work and published the paper.
okay.
So maybe in future we'll try.
Yeah, is your industry or company involved in that? Mane, are they involved in this all of the designing of the drug to the market level, or it is only doing the preliminary work over
Ma'am,
there?
I cannot hear you properly.
Yeah, am I audible, Momita, now?
Yeah, yeah. Now you are audible.
Yes. So, is the human trial as well as the after procedures done by your company on different
No,
drugs?
this is my postdoctoral work.
Okay.
So in my company, it's only synthesis is mainly done.
Okay, okay.
We
So,
only synthesize molecules which were given to us. Means here no biology work is done.
okay. So it is basically the organic synthesis of the compound which you are doing. That is so nice because you are getting in touch with the chemistry always.
Huh.
Yes, and the docking thing, do you do in your company now?
No, no. Now we don't know. These are all postdoctoral works.
Okay, okay. So
Hmm.
you actually synthesize different kinds of drugs there,
Yeah.
and then I think the synthesized drugs are used by the other industries or
Other
companies.
there we synthesize only those molecules. This
Okay,
design and synthesize by this one given by client. Then we
okay.
after synthesis we hand over the molecule to client.
Yes, yes. And I could see different glimpses of the synthesis. It was spectroscopy and other things you do, and then you confirm the compound and then you give to the client.
Yeah,
Right?
yeah.
Okay, now I have understood. Okay, so it was
Okay,
so
so me the
nice.
one thing for in vivo studies, you have done it on animals. Which kind of animal or something for in
Mr.
vivo
Rats.
studies?
Yes, ma'am.
For in vivo studies,
Huh?
uh, you have used some animal or like animal trial or some mammal trial, something like that. In for in vivo studies, what did you do?
For in vivo studies, we have done with Vista rats. Rat.
Rats. Okay,
Rats.
okay. Because that gene of rat are mostly similar to human genes, no?
That's right.
Okay.
Okay, so such a nice lecture and such a warm things we came to know from Moumita. Whenever you come to Durgapur, please visit us, and we will be arranging a lecture in the same room when you were the students. That will be an honor for us, right?
Thank you so much. It is a high honor for me too.
Yes, yes. We want to always be connected to our students as well as our passed out students. So many thanks to all of you for joining, especially to Moumita. For I'm not telling Dr. Moumita Josh because you were
Not
one of our students.
yes, definitely.
So we can just wind up the session, and we are not ending, we are connecting with Momita, and we will always be keeping in touch. Students can ask any questions related to the same to Momita, and we will be communicating the same to her. Okay, so we can just wind up the session. So thanks to all for joining. Many thanks to Momita.
Thank you
Thank
so
you.
much.
Thank you, Moumita. Thank you.
You're
Thanks,
welcome.
everyone.